Mutations to cell membrane flexibility are characteristic of cancer cells. They can lead to cell division failure, aneuploidy, multinucleated cells, as well as entosis, metastasis, and a greater ability to evade immune cell attacks. The protein mechanisms that drive these behaviors are being researched in the model organism Dictyostelium discoideum, an amoeba. When mechanical stress is exerted on a cell cortex, cross-linking and motor proteins can sense this force. The cortex is mostly filled with polymerizing actin. Cortexylin proteins bound to these actin filaments experience tension that creates a catch bond. Filament responds similarly to shear deformation, and alpha-actinin to dilation deformation. Hexameric monomers of non-muscle myosin II, or NM2, form dimers, tetramers, and bipolar thick filaments, their functional unit. Tension can freeze NM2 bipolar thick filament motor proteins in their strongly bonded state. Both catch bonds and strongly bound NM2s drives the accumulation of more of these proteins to share this force and make the cortex more rigid. For these proteins to act efficiently, they are pre-assembled in nearby condensates called contractility kits, CK, that are held together by the glue-like protein discoidin. CKs exist within a spectrum of mechanoresponsive to non-mechanoresponsive states. Scaffold proteins called IQ gaps competitively bind to cortexylin to regulate contractility kit activity. A single or a pair of IQ gap 1s can bind, one of each, or a lone IQ gap 2. IQ gap 1 promotes dissociation from the cortex, IQ gap 2 promotes association with the cortex. There is always a gradient of both. Whichever is present in greater number drives overall behavior. Mechanoresponsive kits disassemble and associate with the cytoskeleton at the cortex when needed, and when the stress subsides, the proteins begin to dissociate from the cortex and reassemble back to their CK states. This research has led to the discovery that the compound 4-HAP causes the accumulation of contractility kit proteins in the cortex, making cancer cells more rigid and preventing metastasis from occurring.